We are extremely honored and grateful to have Dr. Jim Allison and Dr. Padmanee Sharma with us today. The two are not only an exceptional, world-renowned pair of researchers making major advances in immunotherapy, but also a married couple. Dr. Allison and Dr. Sharma have focused their works on the development and application of therapies for cancer that enable the immune system's T cells to target and destroy cancerous tissue. Dr. Allison once said, "It takes a special type of person to be a scientist. Your hypotheses are usually going to be proven wrong, and it takes perseverance. But the big jumps come from fundamental studies of first principles, so the rewards are huge if you take the chance." Luckily, Dr. Allison and Dr. Sharma took that chance, and it certainly led to a huge reward, as the global scientific community's understanding of one of the most mysterious and malicious disorders, cancer, has been greatly advanced thanks to their research. I will now hand it over to Noel.
Among his many honors, Dr. Allison is a member of the National Academies of Science and Medicine and received the Breakthrough Prize in Life Science and the Lasker DeBakey Clinical Medical Research Award. He received the Wolf Prize in Medicine and the Balzan Prize for Immunological Approaches in Cancer Therapy. And most recently, Dr. Allison received the Nobel Prize in Physiology or Medicine with Dr. Tasuku Honjo for his discovery of cancer therapy by inhibition of negative immune regulation. Currently, Dr. Allison is the Regental Professor and Chair of the Department of Immunology, the Olga Keith Weiss Distinguished University Chair for Cancer Research, the Executive Director of the Immunotherapy Platform at MD Anderson Cancer Center at the University of Texas, and the Director of the Parker Institute for Cancer Research. Fun fact, he is also the lead harmonica player for the checkpoints, named after his discovery of ipilimumab, which is the first in a class of drugs known as checkpoint inhibitors. Now I will hand it over to Ardil.
Since 1996, Dr. Sharma has authored 80 scientific publications, which includes her work in comparing T cells in blood and tissue samples in cancer patients. Dr. Sharma is now a professor of genitourinary medical oncology and immunology in the Division of Cancer Medicine and the scientific director for the immunotherapy platform at the University of Texas MD Anderson Cancer Center, as well as the co-director of the Parker Institute for Cancer Immunotherapy. In addition, Dr. Sharma has won numerous awards, including the Doris Duke Clinical Scientist Development Award and the American Society of Clinical Oncologists Young Investigator Award. She also earned the Melanoma Research Alliance Young Investigator Award and the Prostate Cancer Foundation Challenge Award, which supported her work in novel immunotherapy treatments. Most recently, Dr. Sharma was awarded the prestigious William B. Coley Award for Distinguished Research in Tumor Immunology for her work in anti-CTLA for checkpoint immunotherapy, which was the first checkpoint immunotherapy approved by the FDA to drug Yervoy. Please join us in welcoming Dr. Jim Allison and Dr. Padmanee Sharma.
Thank you.
Okay, I guess I begin then. I'm having a hard time with the audio here. Can everybody hear me okay?
Yes, we can hear you.
Okay, I'll go to the presentation. Thank you for that very kind introduction. And today I want to tell you about some of the basic science that we did that led to these findings. But what I'm going to concentrate on is first just a brief introduction to the findings that led us to propose checkpoint blockade as a mechanism of therapy, and then talk about some molecular detail about the mechanisms of the two major checkpoint targets for immunotherapy, which have been used both singly and in combination, CTLA4 and PD-1, and convince you hopefully that these are two very different checkpoints with very different, and their blockage has very different effect on the ensuing immune responses to tumors. A lot of people have contributed to this work over the years. I'll mention most of them as they come along. But most of the work that I'm going to show you on mechanism was done by a postdoc named Spencer Way, and when he was in the lab, with contributions from the others that are shown there. And a lot of the work that you'll hear about was also all done in collaboration with Pam Sharma and some of it with the immunotherapy platform at MD Anderson. So all of this really work stems from fundamental science, not from any understanding of cancer biology, but rather of T cell activation, which is something I've been studying since the early 80s, actually. And basically what we found was there is an antigen receptor on T cells that recognizes antigens on the surface of cells, but that's not sufficient to activate a naive T cell. It requires a second signal, a co-stimulatory signal, it was called, that we showed in 1988 was a molecule called CD28. And when a T cell receives contemporaneous signals from those two receptors on the T cell, provided by antigen on the antigen presenting cell, plus molecules called B7 molecules, only on very specialized kinds of innate immune cells called dendritic cells, can really initiate an immune response. When a T cell gets those two signals, it turns on this GO pathway that turns on cell cycle machinery that lets the cell in growth factors and turns off cell death molecules that leads to T cells to rapidly proliferate and generate numbers. And this is essential to the generation of immune response, because you start off with probably 100 million different T cells, that is T cells with different receptors in your body to cover the whole universe of antigens, of things that might hurt you. And of course, that leaves you with only a few against any individual antigen. And so we recognize the cells have to proliferate and generate a whole army of T cells to go out and deal with whatever the insult is. So that's what CD28 plus CTLA4 starts. But what we showed in the early 90s was there's also a molecule called CTLA4 that's induced. It's a whole off program. With time, CTLA4 accumulates, and we think it outcompetes the B7 molecules for binding to CD28 and eventually stops co-stimulation, and the T cells run down and can no longer proliferate or perhaps even do their effector function. This is an absolute critical step in all immune responses, because you have to stop that proliferative phase. And we showed that along with other labs by the fact that when you knock out, when you disable the CTLA4 gene in mice, they die because they fill up with T cells. They just can't stop dividing. So what does this have to do with cancer? Well, we had shown in the 90s that tumors are really invisible to the immune system, not because they don't have antigens, but because they don't have these ligands for CD28. They cannot provide that second signal. And we showed that by transducing the genes for B7 into tumor cells, and they immediately get rejected. They won't even grow in mice. So they have antigens, but they're invisible because they just can't be co-stimulated. And what that provide co-stimulation, what that means is the only way to get an immune response is by a process called cross-priming, where T cells die, tumor cells die, perhaps because they got too big, can't get enough nutrients to grow or enough oxygen or whatever, but they undergo a necrotic cell death, cause inflammation, and these cells from the innate immune system come in, pick up the tumor cell debris and represent it in the context, cross-present it now with the B7 molecules. And that gives the two signals that I talked about before, and you get proliferation of T cells. But inevitably, that also induces that off program, that second program, which can proceed with time and stop the T cells from proliferating anymore. And so when we realized this, I thought about this and said, well, what this means is it's kind of a race, because the tumors have had a head start. Unlike bacteria or viral infections, which immediately alert the innate immune system, tumor cells, because they're self, they don't provide that signal until they die and they're picked up. But in any event, it's just a race between the tumor cells and the emerging T cell response. If the T cells get there in time to eliminate the whole mass of tumor cells, everything's okay. But if they don't, then the tumor wins. And so I had the idea, and maybe that's why a lot of therapies don't work. But I thought that if we just disable the brakes with an antibody to CTLA4 and just let the T cells keep proliferating as long as they want, maybe we could do something about cancer. And this was compelling for a couple of reasons. One is you're treating the immune system, not the cancer cell. So in theory, the type of cancer shouldn't really matter. This might work against all kinds of cancer. And secondly, since it started by cell death, you could combine this with standard therapies, which kill tumor cells like chemotherapy, radiation, any of the other more conventional therapies, and give them the properties of the immune response. First of all, which is polyvalency, because despite whatever the target of the drug, the tumor cell death and cross-priming will reveal potentially many of the mutated proteins that are found in cells as a result of the carcinogenic process. And secondly, memory, because once you've got T cells, you've got them potentially for the rest of your life in case the tumor reoccurs. So great idea, but did it work? And this is one of our first experiments. This is an experimental tumor. It doesn't really matter what it is, but anyway, it's a common method where you inject these immortal tumor cell lines on the backs of genetically identical mice, and they grow. And after a while, you have to euthanize the mice. If you block CD28, they grow faster, showing that there is a bit of an immune response, but it can't get there in time, especially when you block co-stimulation. But when you add anti-CTLA-4 antibodies, the tumors grow for a while and then are eliminated. And those tumors, those mice will survive a re-challenge with that same tumor for the rest of their lives. They're permanently immune to recurrence of that. And so after a time, to convince a largely skeptical audience, we joined up with a team called Metarex that had mice that they had engineered. So that instead of having mouse antibody genes, they had human, which is important if you're going to make a drug, because you can't inject mouse proteins into humans. They'll be rejected themselves. So we were able to make a fully human antibody to human CTLA-4 in these mice. And then with Metarex and ultimately with a larger company, Bristol Myers Squibb, we went into clinical development. And as we had predicted early on in early trials, there are objective responses in many tumor types, including melanoma, prostate cancer, kidney, bladder. Dr. Sharma will talk to you about her early experience with bladder, even before the approval of this for melanoma. In the mouse studies and in toxicity studies in cynomolgus monkeys required by the FDA, we never saw any adverse events at all. But when we went into people, it turned out there's no free lunch. There are a lot of colitis, hepatitis, hepapocystis, a lot of itises. These are not necessarily, in fact, are not autoimmune syndromes. They are immune inflammation resulting from a dysregulated immune system, but they can be treated with steroids, and then the patients can be weaned off those, and they don't come back. So they're temporary conditions that are generally manageable. However, now we realize there are that hundreds of thousands of people have been treated, genuine and dangerous autoimmunities, including type 1 diabetes in some patients, which may be a result of just exposure of a subclinical problem beforehand, but also myocarditis and in some cases rheumatoid arthritis. But thank goodness these are generally not, except the myocarditis again can be managed. So just to show you from the start how well this can work, this is the first patient I ever met. This is a woman who was 22 years old. Sharon was her name. When she graduated from college in 2004, she was just about to begin her life, get married and start her career when she began feeling tired, etc. Anyway, she was diagnosed with metastatic melanoma. This shows a CAT scan of her chest at the time. She had 2 subcutaneous nodules on her skin, 31 lung mets, some of which are shown here. As I'll show you in a moment, a 0.5 centimeter brain metastasis. This is before therapy. She went on to fail 3 different chemotherapies, a couple of immunotherapies, high dose IL-2, in a vaccine, radiation to a brain tumor, none of which worked. And she was just about hospice bound when she entered a trial and received a single round of treatment with anti-CTLA-4 in the very early stages, way before it was approved. And all her tumors went away. All the lung mets you can see here gone, and also this 0.5 centimeter brain tumor went away. And here she is in 2016 with her 2 children. And we actually visited them, Pam and I visited them a couple of months ago, and those kids are now teenagers, and so she's cured of disease and is now living her life normally, which for a basic scientist was a very gratifying thing to see it come out of the work. Just a moment here. I always have trouble with this. So by 2015, there are sufficient patients. This shows almost 5,000 patients for whom there was 10 years follow-up. What you could see is that the survival curve of people treated with NanoCTLA-4 flattens out at about 3 years and essentially stays there through 10 years in a little over 20% of patients. And many of these are still alive. So these patients can be considered cured. When we started this work, it's worth pointing out that the median survival of patients after diagnosis with late-stage melanoma was 7 months, with fewer than 3% being alive after 5 years. So this was quite a remarkable finding and got a lot of attention. However, looking at it from another viewpoint, why is it just 20%? And there's a lot of reasons for that, a couple of mechanistic ones I could give you, but of course, one of the most obvious ones and more interesting ones scientifically is, well, maybe there are other checkpoints. It turned out this was correct. There was another molecule called program death 1 that had been discovered by Tasuku Honjo, with whom I shared the Nobel Prize, but who teamed up with Gordon Freeman and Eileen Sharp, who showed that, like CTLA-4, it had a couple of ligands and was another immune checkpoint. And it differs, though, in a couple of ways. One of them is that one of its ligands can actually be expressed on tumor cells themselves. It's induced by gamma interferon, but it inhibits effector cells when they're attacking the tumor cells. So this doesn't work at priming at all. It works on fully differentiated cells. Anyway, an early phase 1 trial, it worked very well in melanoma, non-small cell lung cancer, renal cancer. Didn't work in colorectal cancer in a small panel, but after this, it was found that there is a subset of colorectal cancer that has genomic instability, and so a lot of mutations in those patients do respond. I'll come back to that in a minute. But also castrate-resistant prostate cancer did not respond. As you remember, I mentioned that it had responded to CTLA-4. So this difference, Dr. Sharma will be talking about in a few minutes. I think we understand why that is the case, why one approach is effective and the other is not now. So where do we go from here? Well, of course, we're back then. And obviously one thing was, since we have 2 checkpoints that apparently have different mechanisms, put them together. So first we did this in mice, showing here where they're at least additive in a mouse model. But this led to a few clinical trials in melanoma again. And then this just shows the latest report on a phase 3, follow-up of a phase 3 trial, where ipilimumab was given either by itself or with NanoCTLA-4 or with nivolumab, the Pdl1 antibody. And the 5-year survival, and now it's out to 6 years, is slightly over 50%. And there's no reason why this isn't going to go out 10 years like NanoCTLA-4, like ipi alone. So these patients, I think it's safe to say, have also been cured. So just to point out the significance of this again, was this was almost a death sentence in 2011 when ipi was approved. And now a little over half the patients can look forward to, hopefully, essentially normal life after that. And there was a report last week, for the American Cancer Society, that although the incidence of melanoma is going up, the death rate from the disease is going down, and they attributed it, and this has happened since 2011, and they attributed that to these advances in immunotherapy is leading to a decrease in the death rate overall in the US from this disease. So very nice thing to see, and I hope we can do it in more types of cancer. So what's becoming apparent, though, is that many cancers don't respond. And this just shows one of the things that the Genome Project and genome sequencing techniques have allowed us to realize is that cancer basically is caused by mutations, targeted by mutations in genes that control cell cycle and cell death and things like that. But the burden of mutations in different kinds of cancer is different. And this red box here are patients, are cancers which respond to monotherapies, either CTLA4 or PD-1. It turns out they are ones with high mutational loads. That means that they have demonstrably more neoantigens generated by that mutational process. And these include melanoma, which again is mutations caused by ultraviolet radiation, and then all kinds of smoking-related cancers caused by the carcinogens in tobacco. Those cancers, incidentally, have a lot of mutations. They're genomically unstable. They're very hard to treat for those reasons. But paradoxically, that makes them sitting ducks for the immune system because the immune system loves mutations. The more, the better. But if you go down here, there are other tumors that have lower mutational loads. Renal cancer, kidney cancer responds very well, even though it has low levels of mutations and might not be expected to. But yet it responds very well for reasons that aren't completely understood yet. Prostate is also very cold in terms of mutations. It does respond a little bit. And again, you'll hear more from Dr. Sharma about strategies to attack prostate cancer. But some that haven't responded yet very well at all are pancreatic cancer and glioblastoma. And so we've still got some challenges ahead of us, even though, as shown here, there are 40-something some odd approvals by the Food and Drug Administration of these agents, in a variety of cancer indications. The first being anti-CTLA-4 for melanoma in 2011, followed by an anti-PD-1 antibody, nivolumab, in 2014, and then the combination of the two in 2015. But as you can see, also approvals in lung cancer, renal, and it goes on and on and on. Again, many cancers. I'm not going to go through all that, except to say two things. One is the response rates in these cancers is typically 20 to 30%, and we need to get that higher. And also, what I didn't mention is that there's also approval for any kind of cancer, any histotype that has defects in DNA damage and will therefore have higher levels of mutations. But again, absent from this list are things like pancreatic cancer and glioblastoma. We need to work on those. So what I want to now spend the last part of this talk on is talking about the differences between checkpoints, because initially there was a tendency to say, oh, well, you've got these two checkpoints, but it became clear very quickly that adverse events with PD-1, actually I should have mentioned that, they're basically the same in kind, maybe more pneumonitis than colitis or whatever, but it's much less frequent in PD-1 than in anti-CTLA-4. But there are other differences too. As I told you, CTLA-4 is hardwired, that pathway. It's involved in stopping any immune response, no matter how it gets started. PD-1 is actually an induced resistance, as I told you. The tumor cell begins to express PD-1 in response to gamma interferon made by the T cell trying to kill it. So it's an active resistance mechanism. PD-1, incidentally, has been implicated in protecting the developing embryo in the mother from being killed by the mother responding to the father's genes encoding proteins by the father, and which would be immunogenic in the mother. So it helps protect the fetus. And tumors somehow seem to have co-opted that pathway to protect themselves. CTLA-4 seems to target the CD28 pathway, though there's a little bit of controversy about that. But PD-1, the TCR pathway, there's controversy about that, I should say. So I told you there's a lot of data now that CTLA-4 works during priming, whereas PD-1 works at the other end on fully differentiated functional T cells. Because of this, CTLA-4 can recruit new T cells into a response that weren't there before, whereas PD-1 blockade doesn't do that. It just expands cells that are already there. Responses, anyway, go on and on. As I'll show you, CTLA-4 blockade primarily affects CD4s, although CD8s also, whereas PD-1, at least as a monotherapy, only affects CD8 T cells. As Dr. Sharma will show you, at least in the case of prostate cancer, CTLA-4 blockade can move T cells into cold tumors, otherwise cold tumors that aren't infiltrated, whereas PD-1 cannot. And I've talked about adverse events, but the disease recurrence after responses with CTLA-4 are quite rare, actually, but with PD-1, they're significant, up to 25 to 30% after a couple of years. So they're very different mechanistically and potentially differing in durability. So in order to look at this, we exploited a technique called time of flight cytometry. And the details don't really matter that much, but what they involve is taking monoclonal antibodies to specific targets, tagging them with heavy metal ions, labeling a cell, and then sorting them one at a time by flow. And then you can analyze up to 43 different parameters on cells by putting the stained cells one at a time through a mass spec where they're vaporized and the ions counted, which tells you how much of the different antigens were there. And so very powerful technique. And doing this in individual mice and keeping track of the tumor size, we could relate changes in different molecules in the different cell types with different outcomes, with increases or decreases of tumor size. So this is the typical setup. This is a methylclanthin. Anyway, it doesn't matter. But anyway, this is a transplantable tumor, pretty strongly immunogenic. Could liken it to a melanoma situation in humans. So we give the mice tumors, treat them with antibody. At about the time the tumors start to shrink, harvest the tumors and put them through the CyTOF machine. And this is basically what it looks like. We're using software that groups cells according to these 43 parameters, according to what markers they have. And then if we gate on markers, including CD45, which means hematopoietic cell, and then CD3, which is subunit of the T cell receptor, we can look just at T cells. And so the blue dots here in these dot plots represent cells, individual cells in analysis for control treated mice. Red dots are after treatment with anti-CTLA-4, and green with PD-1. And we can clearly see down here with the NKT cells and gamma delta cells, they aren't affected at all. There's a random distribution. CD4 effector cells are mostly red here. That's because they're greatly expanded, as I'll show you, by CTLA-4 treatment. There's Tregs are depleted of those. And this is something that happens in mice, but not humans because of the antibody. And then the CD8, you can see mixtures of green and red patches showing that there are different kinds of CD8 cells that are affected by the two treatments. So to figure out what's what, you can line up these different clusters that are defined by that computational analysis with what's called a phenogram. These are the individual gene products that we're measuring here to get an idea of what's going on. Of course, this is uninterpretable. So we could define these as different cell types based on those genes expressed and annotate them as shown here. And so regulatory T cells, which are cells that, as defined, can inhibit other kinds of T cells, get away with their effector function. As you can see, CTLA-4 tends to deplete one of the subsets, the major subset of regulatory T cells. And again, this happens in mice, but it doesn't happen in humans because of a peculiarity of the antibody in humans. But that's not the only mechanism of action of CTLA-4. But anyway, the number of those cells is associated with bigger tumors. So the more of these, either one of these two types, and the differences in this maturity marker KLRG1, but the numbers of either of those are associated with bigger tumors, as you might expect. Cd4 cells, there are two types, one of which is this odd cell that you'll hear a lot more from Dr. Sharma. It's a CD4 Th1 cell, meaning that makes gamma interferon and TNF alpha, but it's very different because it expresses a molecule called Icos. And I won't go into that anymore, leave that for her to discuss. But those cells are greatly increased by anti-CTLA-4 and not by PD-1. And there's another cluster of cells that looks like naive T cells, and they're not really affected by by any of the treatments. But anyway, these cells, of course, are strongly affected with smaller T cells. There's another population of CD8s, which is expanded by CTLA4 that you can see here. It's a CD8 effector cell, which is also associated with smaller tumor size. So this cell and this cell, are affected by CTLA4 and not PD1. If you look at PD1, it does nothing to CD4s, but it expands with this peculiar set of CD8s here that express not only high levels of PD1, which is an inhibitory molecule checkpoint, but also two other checkpoints, TIM3 and TBET, which are, TIM3 and LAG3, not shown here. TBET is a marker of function. So these are what are called exhausted CD8 cells. It's been known before that PD1, blocking PD1 allows those cells to keep proliferating, whereas otherwise PD1 would stop them. And CTLA4 does not affect those cells. Interesting, these cells still have that phenotype. What that tells us is that if the antibody goes away, those cells are going to stop proliferating, which explains something that I didn't mention before, is that typically CTLA4 is given for only 3 injections max over a period of 3 months, whereas PD1 antibodies are typically given for up to 2 years in treating patients. And this is why, because if you stop treating, those cells are going to stop dividing. And we think we found a way around that I'll show you in a moment. But PD1 also, and those cells are definitely associated with smaller tumors, as are these effector cells that CTLA4 expanded. So just to the other changes that occur are really not relevant to anything. So if you just reduce this to looking at the cell types, CTLA4, there are 2 cells involved in its anti-tumor effect, these CD4 Th1-like effector cells and CD8 effectors. PD1 blockade gives you those effects, not those CD4s, but the same CD8 effector. But also these exhausted cells here, which would otherwise not be effective at all. And so I think in the interest of time, I'm just going to cut to the last point. And that is what happens, that's monotherapy. So what happens when you put these together? In other words, what's happening in those patients, the melanoma patients, for example, that are treated with both antibodies that have such a durable response? What's happening in them? So basically the question is, you treat with anti-CTLA4 and anti-PD1, do you get just the sum of the effects or do you get something new? And so we carried out that same sort of analysis, except we added a 4th group, of course, which is anti-CTLA4 plus PD1 instead of just the monotherapies or the control. And so now if we look at the CD8s, this shows again the results of this one cluster of the CD8 exhausted cells. And what you can see here, the PD1 high, LAG3 high, TIM3 high, that are expanded by PD1, but not by CTLA4. When you add CTLA4, really they drop to below control values. And as we'll show you in a moment, they become irrelevant, whereas they were strongly associated with the anti-tumor response before. Now they become irrelevant. Well, this actually shows the data. So this just shows the more of those exhausted cells, the smaller the tumor. But after you treat with anti-CTLA4 plus PD1, they become irrelevant. So not only do the numbers drop, but they don't matter anymore at all. So the mechanism is not just the sum of the two. And as far as the effect, the PD1 effector cells, the non-exhausted cells that were expanded by both PD1 and CTLA4, you can see that these are expanded much more by the combination, which makes sense because you're making them more easily to co-stimulate because you removed remember that's what CTLA4 does, is prevent co-stimulation, which is needed to sustain an immune response. And so you can give them co-stimulation coming in, but also prevent, allow them to just keep dividing. And if you look at the CD4s, they were enhanced in monotherapy just by CTLA-4 and not PD-1. When you add the combination now, that increases the cells more because activated CD4s also express PD-1. And since they're induced and are by CTLA-4, which I didn't have time to show you, now they express PD-1 and they can also be expanded. So the combination of these two antibodies does not result in just the sum of the two, but leads to expansion of these effector cells and seems to, in these other cells, the exhausted cells go away. So it's just the two effector populations that seem to play a role in that even more robust response that you see in melanoma and the combination therapy. So what happens to them? Well, one possibility, when you give the combination, one possibility is that the presence of anti-CTLA-4 antibodies converts those exhausted cells back to effector cells that aren't exhausted. And we think that's unlikely because there's considerable data now that shows that those exhausted cells have epigenetic changes that are not reversible. And so while that may be possible, it seems highly unlikely. We're looking at the data now to see if that might be by looking at markers that might indicate that they had an epigenetic change that might have been reversed. But the more intriguing possibility to me is that it's prevented those cells never get a chance to get exhausted because they are easily co-stimulated by CD28 because you don't have CTLA-4 around anymore to get in the way of the co-stimulation. And that's why that combination therapy works so well, which suggests that maybe we wouldn't have to give PD-1 antibody for so long. But those are open questions now, but just shows that you need to really look at mechanisms and get data and not just assume that things are going to be, some of what you observed before. So I'm gonna finish now by just saying that I've just talked about these two checkpoints, CTLA-4 and PD-1, but there's many more LAG3, TIM3 I've talked about that go together with PD-1. And cells that express all three of those, the T cells are really in trouble. But there are other ones like LILRB4 is a molecule that we've been studying that's not expressed on tumor cells or bona fide antigen presenting cells, but expressed by a suppressive cell, suppressive type of myeloid cell that you'll hear about Dr. Sharma again in a moment. And they're also positive co-stimulatory molecules like Icos that again you'll hear about next, but also other molecules such as 4-1BB and CD226 and OX40 and other things. So there's a multitude of these molecules. PD-1 and CTLA-4 are probably the major ones, but these others could all contribute to a response. And so there's a lot of work being done to block multiple ones, but also there's beginning to be work done because conventional therapies, including radiation, chemotherapy, and the genomically targeted therapies ultimately kill cells. And they would start an immune response, which would be quenched by these feedback mechanisms that I talked about. But by combining CTLA-4 or PD-1 or these other blockade mechanisms, but in particular PD-1 and CTLA-4, we think you could give these conventional therapies the power of immunotherapy. That's a lot, that work's just beginning, and I look forward to a situation where I think all therapies of clinical cancer are going to involve combinations, including immunotherapies. And hopefully we will be able to cure many more patients with many types of cancer. And so this is the goal that we have, Pam and I together, this is what the cancer therapy, cancer oncology research has looked like for the last 20 years since the advent of chemotherapy. Is you treat a large number of patients with whatever your drug is, and you look for an increase in median survival, which may be weeks or months, which is, if you can survive that with good quality, if you can obtain that with good quality of life, that's certainly worth doing until a new therapy comes along or whatever. But we know from ipilimumab, from CTLA-4, that you can do that. It moved the median survival over about 4 months, but gave this tail of survival at about 20%. And this is a curve that represents our aspiration, not data. But rather than concentrating on the median survival so much, what we think needs to be done is really concentrate on raising that survival tail as high as we can get it in as many different cancer types as we can get it, so that we could cure more cancers and more patients of many types. And so the good news is, I think, that we know it can be done in some patients. And the better news is, through the work that you're going to hear about next, we're beginning to learn the basic rules for this and how to rationally devise strategies. And so with that, I'll close and turn it over to Dr. Sharma, and I guess we take questions at the end. And thank you very much for your patience, and I hope I didn't go over too long. Thank you very much. Over to you.
Sharma, you're muted.
Is that better?
Yes.
Okay, great. Thank you. Thank you so much for the opportunity to speak with you today. As Jim mentioned, I'm going to be speaking more about what we do in the clinic and how we learn from the patients as we take information from clinical trials back to the laboratory and then establish the models to help us really understand the mechanisms of response and resistance to immune checkpoint therapy. So as Jim mentioned, again, anti-CTLA-4 opened an entire new field called immune checkpoint therapy. And clearly, as he discussed, CTLA-4 started the field, and PD-1 and Pdl1 were next in terms of drugs that led to clinical benefit. But as you can see in this cartoon, and as he pointed out, there are many other targets that we need to consider in the immune response that can be used for driving tumor rejection. So the key research questions that remain at this time are: why do some patients respond and others do not? Can we identify biomarkers that predict response or immune-related toxicities? Are there biomarkers to enable patient selection for treatment with monotherapy? So because it has fewer toxicities versus combination therapy, which may be more effective, but also may have higher frequency of toxicities. Can we increase the number of patients who respond? And are there other pathways that can be targeted to improve clinical outcomes? I won't be able to talk about all of these things today, but hopefully I'll give you some ideas as to how we're trying to answer these questions. So for us, it's really about integrating the laboratory and the clinical research. And we do this in what we call a reverse translational approach. As you know, hypothesis testing in the laboratory really consists of mice and preclinical models. But the mice are inbred, the disease is homogeneous, and it gives you the answers you may want to know in terms of knocking in a pathway or knocking out a pathway. So you can test mechanisms very easily. However, since we have over 2,000 and maybe even up to 3,000 clinical trials ongoing with immune checkpoint agents at this time, we're really seeing the concept now where the clinic is outstripping our basic science understanding. And so the clinical data is coming very fast, and we need to then look at the patients, which are polymorphic and disease is heterogeneous in that setting. We need to take the data from the patients and generate some hypotheses from these data, and then go back to the laboratory to design the appropriate model to test the hypotheses. So this is where the reverse translation is coming into play, because we're moving from the clinic to the lab. And hopefully we can identify mechanisms then that allow us then to go back from the lab to the clinic. So this is an iterative process going continuously forward. In order to take advantage of what we're learning in the clinic, we had to rethink clinical trial design in order to obtain the appropriate samples for laboratory studies. So as you know, there are phase 1 safety dose escalation studies, phase 2 efficacy studies, and phase 3 comparison to standard of care. These are the standard clinical trial paradigm. However, in these clinical trial setting of phase 1, phase 2, and phase 3, we're really looking at clinical endpoints without really getting sufficient samples for laboratory studies. So we propose these pre-surgical or tissue-based trials, which we call phase 1A or phase 2A, to integrate into the standard paradigm. And so these earlier trials are 1A and 2A trials. allow us then to maybe gain a clinical signal in a tumor type that we were not thinking of previously. It allows us to have access to samples for biomarker analyses, and it allows us to gain mechanistic insights that we can then test in other models for hypothesis testing. So we initiated this paradigm in 2004, and the first trial was conducted in 2006. This was our protocol 20060080. And I'm pointing all of these dates out because really this occurred prior to any FDA approval of an immune checkpoint agent, which the first one occurred in 2011. And it's important to know that as we have experimental drugs, or as you're thinking about experimental drugs that may go on to help patients, that there has to be some testing in patients in the earlier setting to understand whether or not your drug hit its target and whether or not your drug is having the mechanism being targeted in the way that you expect it to. So having the human immune response studied was very important to us. So we designed the first clinical trial to look at this, and this was the first ever neoadjuvant or pre-surgical clinical trial. Of course, now there are hundreds of them, but this was the first one. And it was also the first trial in bladder cancer patients. And of course, again, there are hundreds of trials now in bladder cancer, but this was the first one. And so we designed this trial in 12 patients, whereby we could give the patients with localized bladder cancer who are already scheduled for surgery, because the treatment of localized bladder cancer is surgery to remove the bladder. We asked these patients to participate in the clinical trial, whereby they can receive 2 doses of the anti-CTLA4 antibody. And then at the time of surgery, we would have access to all of the tumor material, since the entire bladder is being removed, to understand what the impact of anti-CTLA4 was on the immune response within the tumor tissues itself. So the trial served multiple purposes. One, it gave us a clinical signal for safety, because it was the first trial to ever be combined with surgery in the neoadjuvant setting. So can you safely give immune checkpoint agents like anti-CTLA4 with surgery? And we were able to answer that question to say yes. It gave us a clinical signal for efficacy, because it was the first trial ever with immune checkpoint therapy in bladder cancer. And we had 3 patients when their bladders were taken out that there were no tumors that were left behind, indicating that the drug can work for bladder cancer. And so this allowed us to then develop other clinical trials that led to eventual FDA approval of these agents for bladder cancer. So these are important points from a simple 12-patient trial. But importantly, the trial also gave us access to the tumor samples. And here you can compare what a pretreatment biopsy and a post-treatment sample looks like, and really identify what is happening in the human immune response after you treat with anti-CTLA4. And here you can see that within the tumor microenvironment, we're getting infiltration of lymphocytes. And these are now when you stain but with antibodies in an immunohistochemical study is shown here. You can see that these are CD3 positive, CD4 positive, and CD8 positive, indicating that these are all T cell subsets, and they're granzyme positive, meaning that they're activated T cells. And of note, we also have CD20 positive cells in these studies, and these are B cells. And so there's some T cells and B cells that are interacting within the tumor microenvironment. When we looked at the differentially expressed genes between the pretreatment and post-treatment samples, what we saw was that there were these top 10 genes that were identified. And of course, since we had increase in the immune infiltration as a result of treatment, these genes are all related to the immune response. What was surprising to us was that Icos was the top gene, because Icos had not previously been known to have a role in anti-tumor responses. And so just to briefly review, Icos is inducible co-stimulator. It belongs to the same family as CD28 and CTLA4, as shown in this phylogenetic tree here. Its expression has been known to be increased on activated T cells. It has a very diverse role that's been reported. It's associated mostly with follicular helper T cells, which really are in lymph nodes and allow for T and B cell collaboration. But a role in anti-tumor responses had not been established. And so we went back to then look at the tumor samples to ask whether or not our gene expression data was confirmed by protein expression. And to look at protein expression of Icos, we did flow cytometry studies on the tumor samples. And when we looked at the Icos positive staining on CD4 T cells in this flow cytometry plot, you can see in non-malignant tissues, which were the ureters that were taken from the bladder, there were about 13% of these cells, and in untreated tumor tissue, similarly around 16%. However, after treatment with anti-CTLA-4, now you can see a significant increase in this subset, up to about 40% of the cells are now Icos positive. So we wanted to understand the function of these cells because they're increasing within the tumor, but what are they doing? And so again, we had access to the tumor sample so we could get the CD4 T cells and these Icos positive T cells from the tumor. And then we could also look at the tumor and see that the tumor expressed certain antigens. Here, for example, NY-ESO-1 is an antigen. And so if we then incubated these T cells with antigens and peptides like NY-ESO-1 peptides, we asked whether or not these T cells were functional and could they produce cytokines such as interferon gamma and TNF alpha. And you can see here in the presence of the NY-ESO-1 peptides, the T cells are activated and now produce interferon gamma, TNF alpha, and MIP-1 beta, indicating that these were indeed effector T cells that were specific for the tumor. We also wanted to know whether or not these T cells correlated with good outcomes in patients. And there was a clinical trial that was ongoing, the phase 3 clinical trial in patients with metastatic melanoma. And so we collaborated with Judd Wolchuk's group to have access to blood samples that they had collected. And we looked for these Icos positive T cells in the blood samples. And we could see that patients who had an increase over time and sustained an increase in these Icos positive T cells had much better survival as compared to patients who did not. In another clinical trial that we conducted, so this is a trial with anti-CTLA-4, which is a different drug, tremelimumab, plus anti-Pdl1 durvalumab. Again, in patients with localized bladder cancer, and here this is patients who had bladder cancer where they were not eligible for cisplatin chemotherapy because cisplatin is usually the standard of care for these patients. But because these patients had poor renal function or other medical comorbidities, they're not eligible for cisplatin and they have very poor outcome for their disease. And so we wanted to know whether we can give them immunotherapy to treat prior to surgery. And here we saw that patients who had the combination therapy, 37.5% of these patients had pathologic complete responses, meaning these patients had no disease at the time of surgery. And in patients who had very large, bulky disease, these 3D masses, we had 12 patients like this. Even in those patients, we saw 42% of these patients with pathologic complete responses. But again, importantly, what we identified was that there were these biomarkers of response, meaning the patients who had a good response to the combination therapy had these structures called tertiary lymphoid structures. And you can see that these were much higher in the responder patients as compared to non-responder patients. And so what the tertiary lymphoid structures are comprised of are the T and B cells that I mentioned before. And this is a picture showing how these cells come together to form these tertiary lymphoid structures. And the T cells that we found associated with these, when you look, was this increase again in these Icos positive CD4 T cells. So again, showing how these cells are playing a role in leading to anti-tumor responses and tumor rejection. But that was all of the clinical data that we had. And so from the clinical data, we generated the hypothesis that the Icos-Icos ligand pathway is necessary for effective anti-tumor immune responses in the setting of anti-CTLA-4. And now we have to test this hypothesis in the laboratory. And so in the laboratory, we went back to the mouse models, and we could use wild type mice, or we had access now to Icos knockout mice, meaning that these mice did not express Icos, or Icos ligand knockout mice, since Icos ligand is the ligand for the Icos molecule. And so now we had the two knockouts and the wild type. And so we can inoculate the mice with tumors and treat with anti-CTLA-4. And you can see the wild type mice are quite capable of rejecting the tumors, and they have 80 to 90% of the mice surviving long term. However, the Icos knockout mice and the Icos ligand knockout mice both had impaired tumor rejection, and they had poorer survival as compared to the wild type mice. And so we had the hypothesis again was that since the pathway seemed to be important, then we asked, can this pathway be targeted and developed as a combination therapy strategy with anti-CTLA-4 to improve responses? And so in a mouse model, we were able to show that when we targeted Icos and gave anti-CTLA-4, and when we did this in wild type mice as shown here, now you can see that there was significant improvement in the anti-tumor responses in the blue dotted line. If we did the same experiments in Icos knockout mice, of course, we lost the ability to have this response because Icos is a key player in having this response. And so now there are many companies developing antibodies to Icos to try and move this idea that we had in mouse models into the clinic. So I do want to switch gears to a little bit, as Jim mentioned, that most of the tumor types where immune checkpoint agents have been approved have been tumor types like melanoma and bladder cancer and lung cancer. And it is thought that because these tumor types have lots of mutations, that the mutations then serve as antigens for the T cells. And therefore, they have lots of infiltrating T cells. And when you have lots of infiltrating T cells, if you block the inhibitory pathways like CTLA-4 and PD-1, then those T cells can continue to do their jobs. However, tumor types like prostate cancer, pancreatic cancer, and glioblastoma, clinically, they do not respond as well to immune checkpoint therapy. And there have been many clinical trials conducted, but we haven't seen the same kind of responses as we've seen with melanoma, lung, and bladder cancer. And so it's thought that because prostate, glioblastoma, and pancreatic, these have fewer mutations, and because they have fewer mutations, then they may have fewer antigens and therefore fewer T cells that are infiltrating. And therefore, if you have fewer T cells, then the immune checkpoint therapy agents can't really do much because you don't have the T cells to block inhibitory pathways on T cells. So we wanted to know, is it really true that the immune checkpoint agents are not able to elicit T cell responses to the mutations? Because on the one hand, even though they have fewer mutations, they're a cancer. Prostate cancer is still a cancer. Pancreatic cancer is still a cancer. And all cancers have some mutations. And T cells do not require hundreds of mutations. A T cell only requires one antigen in order for a T cell response to occur. So we wanted to ask the question, are the T cells within prostate cancer patients capable of recognizing the mutations that are present in prostate cancer? And so to do this, we had patients with metastatic castration-resistant prostate cancer, again, a small trial with 30 patients, whereby we were able to get the tumors, we were able to sequence the tumors, exome and RNA sequencing, identify the tumor mutations, and at the same time, we're treating patients with anti-CTLA-4. We're taking the T cells from these patients, and then we're asking, can the T cells really respond to these tumor mutations? And so this is just one example shown here. This is patient number 7. This patient had 2 mutations, one in rogue one in nucleotide exchange factor 37, and one in dihydropyrimidase. And you can see that these were single amino acid mutation as shown here. And you can see prior to treatment with anti-CTLA-4 or the pre-IPI time point, there are no T cell recognition of these antigens, either the wild type or the mutated antigen. However, in the post-IPI1, post-IPI2, and post-IPI4 time points, this is after anti-CTLA-4 now, you can see the T cells are quite capable of recognizing the mutated antigen, but not the wild type antigen. And of course, these ELISpot assays, which are measuring interferon gamma production by T cells, are done with positive and negative controls as shown here. So it is possible that T cells are capable of recognizing the mutations in prostate cancer. So if they're capable, then maybe they're not trafficking. Maybe the problem is that T cells cannot infiltrate into prostate tumors, and that's why prostate cancer is not responding to immune checkpoint agents. And so to answer that, we had another clinical trial whereby patients who were scheduled for surgery to remove their prostate could receive 2 doses of anti-CTLA-4 prior to prostatectomy. And here you can see now, when we looked at the pre-treatment sample, prostate tumors really are a cold tumor. They have very few T cells infiltrating in the pre-treatment sample shown here. However, after treatment with anti-CTLA-4, now you can see that there's lots of infiltrating T cells. But we should have been prepared as immunologists because the immune system is very tightly controlled. As Jim mentioned, CTLA-4 was identified as a regulatory mechanism so as to control a T cell response, because T cells require T cell receptor signaling and CD28 signaling to drive the T cells to on. But it's those same two pathways, T cell receptor signaling and CD28, that will lead to increased expression of CTLA-4 to then turn the T cell off. And so whenever you're driving the T cell response in one direction, there will be other mechanisms to try and turn it off, because that's how you maintain homeostasis and balance within the immune response. And that's how you prevent the immune response from really becoming an autoimmune condition, so that it does its job for a period of time, but then it's tightly controlled to stop. So here we were driving the responses within the prostate tumor microenvironment. And what we found was, as we were increasing these T cell infiltration, we were also increasing inhibitory pathways such as Pdl1 and VISTA, as shown here. So this is a gene expression study showing what genes were increased within the tumor microenvironment. But we also wanted to confirm the gene expression data by protein expression. And so we did immunohistochemical studies, as shown here. And you can see prior to treatment with anti-CTLA-4, there is no PD-1 or Pdl1 or VISTA that's expressed. However, after treatment now, you can see increase in PD-1 expression, Pdl1 expression, and VISTA expression. PD-1 and Pdl1 you heard about from Jim, and VISTA is a novel immune checkpoint that was discovered by Randy Noel's group. When we looked for where the Pdl1 was expressed in the prostate tumors from patients, we found that it's the CD8 T cells, the CD68 macrophages, and the tumor cells that were expressing the Pdl1. And each patient in the post-treatment samples are in red dots, and each patient in the pre-treatment samples are in black triangles, as shown here. VISTA also is highly expressed after treatment, as shown in the red dots, and it's predominantly expressed on these myeloid cells, these CD68 positive cells. When we asked why prostate tumors are now having such resistance to immune checkpoint therapy, but melanoma tumors seem to be responding well, what we found was when we compared the myeloid cells in melanoma and prostate tumors, as shown here. Now you can see in myeloid cells that myeloid cells have this M1 gene signature, and the M1 gene signature goes along with myeloid cells that actually participate and help the T cells to reject the tumors. However, prostate cancers have more the M2 gene signature, and those myeloid cells tend to be more suppressive and suppress the T cell responses. And we're still trying to understand why one tumor microenvironment has a signal that would allow for M2 development, while another tumor microenvironment would allow for M1 development. But clearly, we were able to show that VISTA and Pdl1 are very potent inhibitors of human T cell responses, as shown here. If you incubate the T cells in culture and give anti-CD3 to stimulate the T cells, you'll see that both interferon gamma and TNF alpha are produced by the T cells. But if when you culture these T cells, you have Pdl1 or VISTA coated onto the plate, now the T cells are not quite capable of producing as much interferon gamma and TNF alpha. They're very much inhibited. So we discussed with Bristol Myers Squibb that we'd like to conduct a combination clinical trial in patients with prostate cancer. And the first combination we did was anti-CTLA-4 and anti-PD-1. And then we were able to publish that now for the first time. We can see these dramatic regression of prostate tumors. So here's a patient with a PSA up to 500. PSA is a measure of tumor burden. And you can see that this now goes down to undetectable levels, and the patient's CT scans also resolve the disease. However, because we were giving the combination therapy study now, we saw lots more toxicities in the patients. And so we had to expand the study to try and give the agents a different schedule and dose, hopefully to maintain efficacy and minimize the toxicity. So that trial is still ongoing. What we did notice in the combination therapy trial, though, was that the patients who had soft tissue metastases were the patients who did much better. So if they had a metastatic lesion that went to lung or to the lymph node, those patients had a much better response than if they had metastatic disease that went to the bone. And as you may know, patients with prostate cancer predominantly have bone metastases, not soft tissue metastases. And because bone metastases are a predominant area, it may be one of the reasons that patients with prostate cancer are not responding as well to immune checkpoint agents. So we wanted to understand the difference between the microenvironment and the immune response in soft tissue metastases versus bone metastases. And so again, we had access to the tumor material from patients, and we can compare the immune response in soft tissue metastases and bone metastases. And when we looked at the pre and post-treatment samples in soft tissue, what we found was that in the soft tissue after treatment with anti-CTLA-4, there was a significant increase in this Th1 signature. And the Th1 immune response is really responsible for tumor rejection because it is the response associated with interferon gamma and TNF alpha. However, when we looked into bone metastases, we did not see an increase in the Th1 response. We actually saw an increase in Th17 responses, not Th1. And so it was curious to us why the bone was skewing T cells towards a development pathway of Th17, whereas soft tissue was skewing T cells in the right direction for tumor rejection, which is the Th1. So we went back to the laboratory and we set up a mouse model whereby we can inject the prostate cancer into the subcutaneous area of the mice to mimic soft tissue, or we can inject the tumor cells into the bones of the mice to mimic the bone metastases. And then we could treat the mice with anti-CTLA-4 and anti-PD-1 and ask what happens to the immune response. And here you can see again, in the setting of the subcutaneous lesions, the Th1 is what increases in the mice after treatment. But in the bone lesions, we're seeing an increase in Th17. And so we had to do very much looking at what could be the mechanisms to skew a T cell development. And it's well known that actually Th17 development occurs when it has multiple signaling such as IL-6 and TGF-beta, which are cytokines in the microenvironment. And when we looked in the bone microenvironment, so if you look at BM, that's the bone microenvironment, and BMT is bone microenvironment with tumor. You can see even without a tumor, IL-6 levels are very high within the bones. So the bone microenvironment naturally has high IL-6. However, when a tumor is present, the osteoclasts within the bone then lead to high TGF-beta. And it's the two together now, the IL-6 and TGF-beta, that then allow for skewing of T cells so that they now skew more towards a Th17 development. So we asked whether or not we can block TGF-beta and treat the bone lesions in the mice. And you can see now in the green box, for the first time, when we blocked the TGF-beta and gave immune checkpoint therapy, now you can see that there was a decrease in tumor volume. And as we published in this paper, there was also increased survival for these mice. Importantly, what we saw was that there was now expansion of these Th1 Cd4 T cells that occurred, an expansion of CD8 T cell clones within the bones of the mice. And this explains why now the bone lesions were able to be rejected in these mice. And so now we're taking these data back from the mice to write a new clinical trial whereby we can block TGF-beta in combination with immune checkpoint therapy. I just want to conclude on a final mechanism of resistance that we've noted. Again, as you know, CD28 and T cell receptor leads to turning the T cell on. But it's also, again, the same pathways that can lead to increase in epigenetic mechanisms such as EZH2. And EZH2 regulates other genes such as FOXP3. And FOXP3 is a transcription factor that allows for regulatory T cells that can now suppress an immune response. So you can see again how the on signals, CD28 and T cell receptor, also leads to mechanisms of off signals such as FOXP3 stabilization, because that will then suppress an immune response. So this yin and the yang of the immune response is something to always keep in mind. So what we showed that in patients who did not respond to anti-CTLA-4, they had an increase in this EZH2 expression within their CD4 T cells. And so we wondered whether or not we can convert these FOXP3 positive cells into effector cells by using an EZH2 inhibitor. And so we went back to a mouse model, and this is a GFP mouse model where we can sort the FOXP3 cells by GFP expression. And then we can incubate these GFP positive cells with EZH2 inhibitor or not, and we can look for their phenotype and their function. And what we saw was that phenotypically we decreased FOXP3 expression and we increased these Th1 related genes such as CXCL10, interferon gamma, and granzyme B. But functionally also these T cells, which were quite capable of suppressing effector T cells in this CFSE dilution analysis showed here, now those T cells were no longer capable of suppressing effector T cells, meaning that we've now changed their function. And in a mouse model, we asked whether or not when we gave anti-CTLA-4 and used an EZH2 inhibitor, could we improve anti-tumor responses in the mice? And what we saw was that when you gave the combination of an EZH2 inhibitor and anti-CTLA-4, you decreased the tumor volume and you improved the survival of the mice, as shown here. And so this led us to write a new clinical trial, which is now accruing patients. And so in this clinical trial, actually, we have now 7 patients treated to date. We have patients with prostate cancer, bladder cancer, and renal cell carcinoma, and they're getting this EZH1/2 inhibitor plus anti-CTLA-4. So hopefully we'll give you an update on that data from those trials, but I think it's nice to see how we can move between the lab and the clinic to then impact new treatments for patients. So I'll just end here to say, as Jim mentioned, there are many immunotherapy targets to consider, where they function, how they function, and at what part of the immune response they function becomes important. So there's lots of work to still be done, and we're at the tip of the iceberg as we move the field forward. So in conclusion, immune checkpoint therapy has certainly joined the ranks of surgery, radiation, and chemotherapy as a pillar of cancer treatment, and combination strategies are the future. Multiple immune checkpoints exist and are dynamic in their expression. Therefore, they have to be evaluated in both pre and on-treatment human tumor samples to guide therapeutic decisions. The organ-specific tumor microenvironment will need to be considered in order to understand immunologic subsets and subsequent immune responses against cancer cells in these organs. Pre-surgical and tissue-based clinical trials provide a feasible platform to study biologic effects in patients. I hope I've convinced you that small clinical trials, 10 to 20 patients, can really provide a lot of data so that you can gain insights into mechanisms and then recognize what mechanisms need to be targeted in order to move forward with rational combination therapies in larger groups of patients. So I have many people to thank. Their names are listed here, but most importantly, I do want to thank the patients who participate in the clinical trial and allow us to have access to the samples for the laboratory studies. So thank you so much, and I'm happy to take questions.
Thank you both for your illuminating presentations. How about a round of digital applause by reaction or just clapping where you are for Dr. Allison and Dr. Sharma? With that, I'm going to turn it over to Alvin, Senna, and Akul, who are going to moderate the Q&A. And just a reminder that we will close the Zoom session promptly at 12:30. Thank you, everyone.
Thank you so much, Dr. Sharma and Dr. Allison, for this amazing lecture. And thanks, everyone, for submitting questions. We had to pick only some of them because we have time constraints, but we tried our best to include the most frequently asked questions. So our first question is, we see that receptors differ based on the type of cell. So why would they not differ based on the type of cancer? And if they do not differ on the type of cancer, is the MHC protein the immune system uses unique to cancers?
So this is a great question. And as you know, what we're studying really is about targeting the immune system, not really the cancers. So the immune system, because of where these receptors are, they're ubiquitous. They're with the same immune system you have, the same immune system I have. We all have these receptors on our immune cells. And they're there to really do multiple function, right? So they're there also to drive an immune response against viral antigens, bacterial antigens, and cancer antigens as well when they can be recognized. And the receptors also control the immune response so that at no point does that immune response take off to become an autoimmune event. So it's that's why the immune cells, whether it's a bladder cancer, a lung cancer, or melanoma, we're still talking about the same immune system. And so, yes, we do have to consider what the tumor cells may also bring to that, but the immune cells have to be what you're thinking about from going from tumor type to tumor type. There may be differences in tumor microenvironment, as I mentioned, but the immune cells do have a lot of the same receptors regardless of the tumor type that we're looking at.
Our next question comes from another participant. They said, what do you think about alternate treatments? Recently, I, or the participant, have been hearing talks about using nanobots to treat cancer. Do you think that would be a potentially effective treatment?
I think there are lots of great new treatments that are going to be coming down the line for cancer, right? I mean, engineering now is becoming the way to move forward with some of the new technology coming into the clinic. So if you think about it, CAR T cells, for example, those are engineered T cells that are now being given. I think nanoparticles and those kinds of things will also be developed and tested. Of course, we're not going to know which ones will have FDA approval and really lead to clinical benefit until they're tested in clinical trials. But I encourage all of the new ideas and new technologies because this is how we move forward to getting more cures for our patients.
I have no audio.
Thank you, Dr. Sharma. And we have another question from one of the participants on, can immunotherapy become an individualized treatment for different patients?
Yes, I think that we can get to individualized treatments because there are things such as neoantigens. And as I showed, there were these mutations that can be found that the tumor cells have these mutations, and maybe the mutations that one patient has is not the same mutations that another patient has. And so then that can become a neoantigen or a new antigen that's specific to that patient, and maybe developing a vaccine or a therapy that can really recognize just that specific mutation will allow then for that personalized therapy. So we can have personalized therapies in that way. We can also have personalized therapies if we learn to identify mutations that multiple patients have. So for example, mutations such as ARID1A or mutations such as P53, these are common mutations, or KRAS, across different tumor types. And maybe we can say it's personalized for that specific mutation. There was an FDA approval recently that said that if you had microsatellite unstable, which again is another way of defining the mutations, meaning they have lots of mutations within the tumors, those patients can now have immune checkpoint therapy. So these are personalized based on specific things that we can define.
Thank you so much. The next question is, how would checkpoint-based drugs work in a patient that is immunocompromised? For example, how would a patient with AIDS respond to an anti-CTLA-4 drug as the CD4 plus T cell is suppressed in these patients?
That's a very good question. You're right. And because the CD4 T cells are suppressed, it may not work in the same way in a patient with HIV. So, and again, it depends, right? Because there's so many great antiretroviral medications out there that these patients now are living quite normal lives with normal immune cell subset counts. So maybe in that setting, you could be able to treat the patient with an immune checkpoint agent. But I think if they don't have the immune cell subset, then of course that would make it difficult for the immune checkpoint agent to have an effect. I'm going to try and move my camera here so that Jim's in, because he's having difficulties with his computer.
I have no audio. I can't hear anything.
Mhm.
Okay, thank you for that answer. Our next question is, what do you think the difference is, or for someone that's interested in oncology, what do you think the difference is for between clinical research and practicing medicine?
Yeah, I think, practicing medicine really comes down to taking care of your patients, knowing what the standard of care regimen are, and making sure that you're giving them the best available standard of care. Clinical research means that you know what the standard of care is, but you want to push the envelope because it's not good enough yet. We're still not curing all the patients that we want to cure. And so bringing back the questions that you want to ask, and make sure that you're asking that question in a research setting to say, can I try a new agent in a clinical trial, or can I understand the mechanism of how something is not working and come back to the clinic with new ideas. So I think those are the difference. And I mean, we need both. Clearly, we need physicians who are well-versed in what the standard of care is so that they can treat patients all over. And we need clinicians at academic centers who are interested in research and conducting the next clinical trial to test an idea.
Thank you. And going back to a question on the research data, one of the participants asked, why is disease recurrence more common with PD-1 drugs? And is there a preferential use of ipilimumab or nivolumab depending on the cancer type?
Well, I think in terms of the differences, it may be that the way they work. Again, NSCLA4 works at priming, and we've got some data that we're just about to submit for publication where we show that it does a better job of amplifying cells with more of a self-renewing capability than the more terminally differentiated cells that PD-1 works at. So inherently you get better memory. Now, there's a lot more than memory to a durability of a clinical response, I should add. So we're not sure that that's it, but that may be part of it.
Yeah, and I think right now what we've seen though is that there are more FDA approvals with anti-PD-1 drugs than there are with the anti-CTLA4 drugs. Partly because, you know, the anti-PD-1 drugs are by multiple companies. So you have a lot more clinical trials with those agents. Also, the anti-PD-1 drugs seem to have fewer toxicities associated with them as compared to the anti-CTLA4 drugs. But we need the two together because they have very different mechanism of action, as you heard from Jim's talk. And so I think the two together will be important.
Yeah, I think the two together are going to become the core of things going on. In various ratios, depending on, you know, the kind of cancer.
Thank you so much. The next question is, since the anti-CTLA4 and anti-PD-1 have almost opposite effects and functions, did the checkpoints serve as reversing effects? Did the anti-CTLA4 function to induce a molecular signal from CD28 to trigger a response after the anti-PD-1 induced resistance and targeted the TCR pathway?
Well, they don't reverse each other because they do completely different things. I'm just saying that when you start out, you've got a mixture of cells that have already been activated and some that are naive. But CTLA4, you know, works on CD28, which happens only during that initial contact of a CD4 cell with a dendritic cell during the initiation of the immune response. So that's when it can work, or later if an exhausted T cell needs to be co-stimulated. But PD-1 is on effector cells, and it can work at any time. But molecular signaling, there's still some controversy about that. Our data, cumulative over the years, suggests that CTLA4 works just by, it's got about 1,000-fold higher avidity for the B7 molecules than CD28. And consistent with that, we've got data that it's just out competing CD28, I should say, for binding the B7 molecules. So it just shuts off co-stimulation, which PD-1 has nothing to do with co-stimulation at all.
And I should point out that even though patients may be resistant to anti-PD-1 therapy, they can still get anti-CTLA4 therapy and respond. And even if they're resistant to anti-CTLA4 therapy, they can still get anti-PD-1 therapy and respond.
Thank you for that distinction. Our next question is, when bolstering the immune response by using checkpoint-based drugs, what is the possible effect on allergic reactions? Would it cause more allergic reactions as the immune system is supported to be more active in how it responds to perceived invaders?
Yeah.
I don't think we have enough data to say that there are more allergic reactions. We have treated patients, for example, with some autoimmune diseases that are well controlled, meaning that they're on medications for their autoimmune disease, and we're able to give the treatment and not see any flare up of the autoimmune disease. So I would expect that in some patients you can give the treatment and not get any flare up of their allergic responses. But again, you have to see this patient by patient and make decisions on a patient by patient basis.
Thank you. And the next question is finding a break for the immune system a sort of requirement to tumor development? And are immunocompromised people or patients more vulnerable to tumors?
Yeah, so it does seem that if you have immune evasion, that's what we tend to call it, right? So over time, as the tumor is developing, your immune system circulating in your body, getting rid of all the mutated cells that occur in your body all the time. But let's say, for example, as those cells are mutating, they've learned how to now, they've come up with a mutation whereby they can evade the immune response. They've knocked out interferon gamma receptor, or they've knocked out class 1 MHC, or then they can evade the immune response, and that can lead to tumor cell development with a larger cancer over time. So yes, that can definitely happen. And people who are immunocompromised are at higher risk and should be screened carefully for cancers.
Yeah. On the other hand, there's a lot of really strong data, particularly in colon cancers and some liver cancers, that chronic inflammation, immune responses, they're not the same immune responses we're talking about in anti-tumor, but anyway, a chronic inflammatory response can actually cause cancer.
Mm-hmm.
So it can work both ways, actually.
Thank you so much. The next question is a personal one. Someone asked, do you believe in luck and science?
I believe in opportunities. I don't know about luck, and maybe the two words are interchangeable. I think if you're presented with an opportunity, you should take full advantage of it. And if you're the lucky one that gets presented with that opportunity, then fully take advantage of it. I know in my career, when I was working in Lloyd Old's lab doing my postdoctoral work, that was when Jim was first proposing this idea about anti-CTLA-4. And I think he proposed it to multiple companies to work with him, and nobody wanted to work with him because nobody believed that, immunotherapy would work for the treatment of cancer. And so Lloyd was one of the people, he was my mentor at the time, but he was one of the people who thought that this idea would be phenomenal. And he asked me to get involved. And that's how I got involved. It was an opportunity that was presented to me as a postdoc. And then I took that and ran with it for my whole career, basically. So I guess I was lucky, but I think it's just more about you have the opportunity presented to you and you should take full advantage of it.
Yeah, there may be something there, and if you don't grab it, it doesn't matter if it appeared or not. I mean, that's what it comes down to. So I don't know what luck is, except an opportunity, as Pam says.
Moving away from the research questions or research-oriented questions, somebody wanted to know, do you have any advice for students who wish to pursue medicine or medical research?
Yes. One, make sure that you have a lot of passion because it's a long road and you want to be passionate the whole way through, right? Make sure you really love it. You're excited by it. You want to get up in the morning and ask these kinds of questions because it takes time before you may get to the point where you see the results of your work, and you don't want to be in a situation where you just feel like you're putting all this energy and getting nothing back, because that may happen for a while, but your passion will keep you going. Yeah. So if you're passionate about it, just keep pursuing it. There are lots of different things to do in science. It doesn't just have to be medicine too. It can, be research. It can be working in pharma industry. It can be lots of different ways to be involved in STEM.
Yeah.
So we have another question, and it's, what is your favorite question to answer, or what has been your favorite question thus so far?
Oh my goodness. I think they're all great to answer. We've had so many interviews over the years, and I think the fun ones are always the ones about mentoring students and, encouraging students for their passion. But I can't pick one favorite one, to be honest. I think they're all interesting. I think the other good ones I've always had is about how do women in science succeed, because the field is very much male dominated, and you have to make sure you understand how women in science need to be supported. So I think those are the kinds of questions, we try to make sure we give answers to that are helpful.
This one was asked very frequently. People want to know what the biggest obstacle has been during your research.
I think for Jim, you could speak, but it was more people not believing in the idea that immunotherapy could work for cancer.
It's by people that they don't want to entertain new ideas, and I think that's rampant in the pharma industry. You follow the trends, whatever people are doing, you do it too. If somebody comes along and says, I got something totally new, they go, wait a minute, I'm not going to step out on that. That's too risky, and consequently I lost about 3 years because nobody would listen to, nobody would I had all these data in mice And people would say, I said, this will work in humans. They said, well, it never has worked before. I said, nobody tried this before. You're just lumping everything under immunotherapy. But we know exactly what this is based on, a molecular mechanism. It's not some, the earlier work that failed was when people didn't know really what antigens were, didn't know what regulated the immune response, didn't know how to vaccinate, didn't know, and those things failed. They failed because we didn't know enough. But but mine didn't come from just saying, oh, I did something that worked once, so I'm going to. We did all the mechanistic stuff and then turned that in. So we did it the other way around. I made sure that I understood the science before I tried to use it, instead of trying to do something and then figure out how it worked, which I think is a very bad way of doing things. I'd rather do an experimental, logical, data-based approach than just some empirical thing. Because if you do it that way, you're likely to run into rabbit holes, that don't go anywhere. And good science, there's no substitute.
Well, on behalf of SSP, we want to thank you, Dr. Sharma, Dr. Allison, for your dedication to world-class research.